Hi, good morning, everyone. So it is my pleasure to welcome you back to our second trainee research grand rounds. For those of you that weren't here last month when we had two of our fantastic trainees present, I wanted to give you just a sense of just how special this is and to really give an opportunity for recognition and celebration of the trainees that will be presenting today. I am one of the co-chairs of the selection committee. Dr. Susan Vogelmeyer is our other co-chair, and this year we had a record number of applications from across our psychiatry and psychology research training programs. I also wanted to thank a couple of the members of the selection committee, including Andy Crystal, Jim Sorensen, Steve Hinshaw, who have also helped to really get these two individuals here today. So with that, I'm going to introduce our first mentor, Dr. Dale McNeil, who will introduce our first speaker.
Good morning. My name is Dale McNeil. Excuse me. I direct the UCSF Clinical Psychology Training Program. And Kaitlin Casaletto is a postdoctoral fellow in our program. Her mentor is Dr. Joel Kramer, who unfortunately is not able to be with us today, so I'm introducing her. Kaitlin received her BA in psychology with a minor in neuroscience at Wake Forest University. She then obtained her MS and PhD in clinical psychology at the San Diego State University, University of California, San Diego Joint Doctoral Program. She completed her internship here in the 2-year UCSF Clinical Psychology Training Program. During her postdoctoral year, she's working in the Memory and Aging Center in the Department of Neurology. Kaitlin has received numerous academic honors and awards and obtained several research grants. She was principal investigator on a 3-year NRSA F31 award from NIDA and currently is the principal investigator on UCSF Alzheimer's Disease Research Center Pilot Award. Kaitlin has published 45 peer-reviewed research articles, 4 book chapters, and delivered more than 50 presentations at scientific meetings. Substantively, during her doctoral work at UC San Diego, she developed cognitive models of everyday functioning to help create behavioral interventions to mitigate neurocognitive disorders in HIV-affected populations. As a fellow in the UCSF Memory and Aging Center, she's applied her training in clinical neuropsychology and cognitive interventions to promote brain health in aging and age-related neurodegeneration. She's particularly interested in identifying potential neurobiological targets that can be modified using behaviors to increase cognitive resilience in aging. It's an honor to welcome Kaitlin Casaletto for her Trainee Research Award presentation.
All right, thank you so much for having me here today, and thank you for that introduction. So I'll be talking a bit about synaptic functioning in cognitive aging and where it diverges from Alzheimer's disease. So first we'll start with, what is normal cognitive aging? I think a lot of us have concepts of what it might be, and patients ask us questions about what's normal as we age. So I thought I would just put this out here first. And these are actual real data that we have published using the NIH Toolbox Cognition Battery, and it's a composite score reflecting memory, attention, processing speed, executive functions. And we can see the normal developmental trajectory of cognition as we age. In really young childhood, we have rapid development of these skills. They kind of peak around age 30 or so, and then we see this sort of gradual decline with age. And this is what we consider normal. And this diverges from Alzheimer's disease, where we see a much steeper decline, probably beginning at least in our 40s, and much more rapidly declining, typically presenting around age 65, and this dotted line is meant to indicate impairment. And so that's really the difference between what we consider normal versus pathological aging. And I thought I would also just briefly touch on Alzheimer's disease in general, just so we're all on the same page, and give some context about how we think about it at the Memory and Aging Center. I think the field is sort of growing to think about Alzheimer's as more of a lifespan disease, almost developmental, such that we think the prodrome is at least lasts for 5 decades, if not more. And there's some really nice work, recent work by Brock, if you're familiar with the Brock staging. This group took a population-based sample of over 2,000 brains post-mortem from ages 0 to 100 and measured the 2 primary proteins that we see in Alzheimer's disease, so tau, tangles, and amyloid plaques, and just measured the levels of these proteins in the brains. And you can see there's actually substantial, the tau is up here in blue, substantial pre-tangle pathology occurring at ages even 0 to 10, increasing in up till the 20s, and then the more pathological stages, the neurofibrillary tangles, which are what we associate with Alzheimer's disease, begin to come on board, at least age 30 and sort of exponentially increasing. And less so, but also in amyloid, we see development of amyloid at least by age 40. And again, this is much different than the typical onset of Alzheimer's disease at age 65, so much earlier developmental progression. So maybe Alzheimer's could be considered not necessarily this age-dependent discrete event, but this slow, very slow progressive disease that extends into older age. And so this is how I've been thinking about cognitive aging as a spectrum into Alzheimer's disease. It's not quite so clear, but more of a gradation. And so what's happening in the normal brain as it ages that could make it vulnerable to some of these pathological misfolding proteins? And this is where I've begun to think a bit more about synapses specifically. So I think everyone here knows synapses are incredibly important. They're the connecting pieces of the brain. They facilitate long-term potentiation, new learning, and memory. We also know that they're highly vulnerable and susceptible in aging. And so actually those cognitive declines that we see in aging are associated with subtle changes in synaptic physiology and not necessarily neural loss in aging. This is a really nice review by Morrison and Baxter, and they show in a non-human primate, a young neuron compared to an aged neuron. And you can see this, the neuron itself, the cell body, is intact. Actually, the dendritic branches are all there. But once you sort of zoom in into the dendritic spines where the synapses live, you can see much reduced synaptic density on the aged brain. And again, this is really linking directly to cognitive function and aging. And also in Alzheimer's disease, we see that synaptic loss actually may precede any of the amyloid or tau aggregation, and is more strongly associated with cognitive symptoms in Alzheimer's disease than either amyloid, which is not associated with cognitive symptoms in Alzheimer's disease, or even tau. So there's something really specific as a biological marker of cognition when we're thinking about synapses. And so until recently, we couldn't measure synapses clinically in humans, in vivo at least. We were relying largely on animal models and post-mortem data. But in the last 2 years or so, some novel markers in the cerebral spinal fluid have been developed where we can begin to sort of get a window into what's going on in the synaptic functioning in vivo. So I'm going to be focusing on neurogranin, which is one of these markers. And so neurogranin is a neural-specific protein. It's found in the postsynaptic dendritic spines. It's a calcium buffering protein, largely found in the hippocampi and basal forebrain. And the way that it functions is in the postsynaptic neuron here, when there's influxes of calcium, Neurogranin releases calmodium in order to basically further potentiate the calcium influx, facilitate long-term potentiation and learning. So it's a critically important postsynaptic protein. And in fact, we see in animal models, if you knock down neurogranin, there's impaired long-term potentiation and memory, whereas if you increase neurogranin, there's increased long-term potentiation and memory abilities. And actually, we've known from postmortem studies that neurogranin protein decreases in the brain in aging as well as in Alzheimer's disease. And now there have been a handful of studies that have begun to look at the CSF marker in vivo and have begun to show that neurogranin is significantly increased in the CSF in Alzheimer's disease. And these studies have begun to show that the CSF levels of neurogranin are actually associated with cognitive performance on these sort of global cognitive screens in Alzheimer's disease. And specifically, I just actually wanted to highlight one of these studies that I think is particularly relevant, that showed basically in people with mild cognitive impairment, which we think about as sort of the prodrome into Alzheimer's disease, those who had the highest levels of CSF neurogranin, so these red and yellow lines here, across a 10-year period showed the steepest decline on cognitive screeners, so the MMSE and the ADAS, and also showed the steepest hippocampal atrophy declines here, and as well as cortical glucose metabolism by FDG PET across time. So thinking that synaptic dysfunction, particularly neurogranin, might be preceding even some of the neurodegeneration that we're seeing in Alzheimer's disease. And so maybe it could give us a window into the biology of what's happening in cognitive aging. And so none of the studies have really characterized neurogranin in normal cognitive aging and tried to try and better understand what's happening at the synaptic level in cognitive aging. And so that was really our aim. Okay. So that was our aim in this study here. It's really characterize what's going on with neurogranin, how does it relate to cognition in a more nuanced way, so specifically looking at memory, given that memory is an important marker of Alzheimer's disease, and how does this relate to the development of these sort of traditional Alzheimer's-related biomarkers. So we expected that neurogranin levels would be associated with older age and worse memory, and that they would probably interact with the Alzheimer-related biomarkers to confer a greater risk of poor memory abilities. So the participants we drew were 132 community-dwelling older adults. They're within normal standards on neurologic and neuropsychological exams, and they were actually drawn through a collaboration at the University of Wisconsin through their Alzheimer's Disease Research Center. And as part of that center, they selected for folks who had an enriched family history of Alzheimer's disease. So these are a subset of which are really at risk for Alzheimer's disease specifically. So you can see, 72% of this group had an immediate family history of Alzheimer's, so a parent or a sibling, and a substantially increased rate of the ApoE4 allele, which is an Alzheimer's risk polymorphism. Sort of base rates are normally around 14%. So that's important context. And we measured neurogranin and cerebral spinal fluid, as well as several of the sort of classic traditional Alzheimer hallmarks. So CSF levels of amyloid, CSF levels of total tau, and specifically phosphorylated tau, which is basically the bad type of tau that we think about in Alzheimer's, as well as bilateral hippocampi, which are the early hit brain structures in Alzheimer's disease. And our primary outcome was a performance on a memory measure. It's called the Auditory Verbal Learning Test. Here, participants had to learn a list of words and basically recall the list of words after a 20 to 30-minute delay. And that delayed recall score is the score that we used in this study. And I also just wanted to highlight how normal these people were. So this memory score, the scaled score, is adjusted for age. A normal score would be 10, a standard deviation of 3. And this group on average performed at 11, so even a little bit better than normal. So these are really what we consider clinically normal individuals. So first, we see that as CSF levels of neurogranin increase, it's associated with older age. This has been shown in postmortem studies, but nice to replicate and see increasing with age here. And in relation to other Alzheimer biomarkers, we see that as higher levels of neurogranin are actually associated with a quadratic increase in total tau, so a really exponential increase in tau, and specifically also phosphorylated tau, so higher levels of neurogranin and higher levels of tau. And to a lesser extent, but also higher levels of neurogranin were associated with smaller hippocampal volumes. So definitely seeing some associations with some of the traditional Alzheimer's biomarkers, but it wasn't associated with amyloid levels in the CSF or family history of Alzheimer's or ApoE4 allele. And so really our question of interest, how do these markers relate to memory, and how can we use them as a marker of cognitive abilities in normal individuals? And we see that actually only neurogranin was the only central nervous system marker that was associated with memory in these normal aging adults. And specifically, if we delve into that relationship a little bit more closely, again we see a bit of a quadratic relationship, such that those with the lowest levels of CSF neurogranin and those with the highest levels of CSF neurogranin show the best memory performances. And this association here held despite controlling for demographics, ApoE4 allele, family history, and all of the other Alzheimer's biomarkers, so a really robust association. And in fact, we didn't see any interactions between neurogranin and each of the Alzheimer's biomarkers to predict memory. So it's not that a subset of these individuals who are sort of developing Alzheimer's are driving this effect. But as you can sort of see here, this is a new biomarker. We want to show all the data possible. But it seems as though there's sort of a group of 6 individuals that are driving this quadratic effect. So we also looked at if we exclude those folks, we still continue to see a significant but just a negative linear relationship between memory and CSF neurogranin. And even if we include those 6 individuals down here and use more clinical cutoff scores, so we use tertials. Actually still the people with the lowest CSF neurogranin showed significantly better memory performances compared to individuals with medium or high levels of neurogranin. So there's a definite association happening between this synaptic marker and memory performance in cognitive aging. So just to recap, so neurogranin was the only CNS marker associated with memory in these normal aging adults. So this could suggest potentially that synaptic dynamics are really highly sensitive to even normal and nuanced cognitive functioning in aging. Synaptic dysfunction might actually precede or act independently from early Alzheimer's pathogenesis, given that these are some folks who are actually at risk for Alzheimer's disease, and we're still seeing this independent association between the synapses and memory. And perhaps monitoring synaptic status via neurogranin or some of the other really exciting protein markers that are coming out may be a really valid and useful marker of age-related brain health, even in the absence of pathology. And so just going into a little bit more detail to try and interpret what's going on. So it seems as though the relationship between neurogranin and memory is a bit nuanced. So as I mentioned, there was sort of the subset of folks who seem to be driving the quadratic effect. So the nature of the relationship isn't completely clear to us yet, given this is really the first study to characterize any sort of polynomic relationship with this marker. All of the other studies have used non-parametric statistics, so using like a Spearman's row, and really not explored possible curvature. So basically, if we're to interpret this, those with the lowest memory, lowest levels of CSF neurogranin had the best memory, and those with the highest level had the best memory. So in low levels of CSF neurogranin, it might be easy to kind of conjecture that low levels in the CSF could indicate high levels of neurogranin availability in the brain. So perhaps in the brain, there's more neurogranin available to sequester this calmodulum in the synapse and further potentiate long-term potentiation. And that sort of makes sense. And then what we're still struggling to interpret, and I would love to get feedback on, is these folks who have the very high levels of neurogranin and better memory. So, maybe it's that this is a cross-sectional study. We're capturing individuals who at baseline have always had low levels of neurogranin, and perhaps this is reflecting more of an efficiency, where these basal levels of neurogranin that aren't necessarily need to be high to produce better memory scores, something about them at baseline. And in fact, these 6 people had high cognitive performances across all of the measures that we looked at. So perhaps these do reflect some sort of successful aging individuals who have sort of a different biological baseline. And in terms of its relationship with other Alzheimer biomarkers, so we saw that neurogranin really wasn't associated with amyloid. And this is actually consistent with most of the previous studies that have been done so far in CSF neurogranin, where they really see very variable or weak associations between neurogranin and amyloid. If they do see associations, it's usually in people with Alzheimer's dementia, so much more progressed disease. And when we think about it, amyloid is an extracellular protein aggregating outside of the cell, and it's actually not associated with cognition in Alzheimer's disease. So perhaps it's that more elevated or pathologic levels of amyloid are necessary in order to interact with more synaptic proteins to predict cognitive performance. And perhaps neurogranin is just a broader marker of brain aging, not necessarily specific to Alzheimer's disease. And on the other hand, we did see that neurogranin was associated with tau levels and hippocampi volumes, and this is consistent with previous studies that have shown actually quite strong associations between neurogranin and tau, in particular levels of 0.6, 0.7 correlations. And this makes a little bit more sense, I think, to me at least. So tau is an axonal protein. It helps provide structure to the axon. Neurogranin is also within the neuron itself, and hippocampi volumes are really just an indicator of structure of the brain tissue, and both of hippocampi and tau are associated with cognition in Alzheimer's disease, and so perhaps all three are sort of these concurrent markers of sort of neural development, neural aging that are coming online together. And so just to wrap up, it's in the early stages, but perhaps something like neurogranin could be used as a biomarker of healthy versus unhealthy cognitive aging. These markers are greatly needed in order to identify and track individuals who might be at risk for neurodegenerative diseases such as Alzheimer's disease. We have a lot of great prevention treatments that are coming online that could be implemented at a much earlier age if we knew something was going wrong in the brain. And so using these markers to really leverage some of the prevention treatments that are coming up. Future works, I think, are really warranted now to better characterize in vivo clinical synaptic aging. I think this could be a really exciting line of research to understand normal cognitive aging, and again, identify plastic targets for intervention. Perhaps even use of these markers in clinical trials as, again, a more clinically important and nuanced marker of the disease state. So a lot of the Alzheimer trials have focused on amyloid. Amyloid is not associated with cognition. Lowering it doesn't seem to be doing much. So maybe more targeted trials targeting the synapses or using synaptic markers as a way to understand, is the trial working? Could be an exciting future direction as well. So I just wanted to thank everyone who's involved, my co-authors and collaborators on the project, especially my mentor, Joel Kramer, who's unfortunately presenting at our Alzheimer's Center renewal today, but also the folks at the University of Wisconsin and Sweden who helped make this study possible, our participants and their family members, and our study funding. And I'd be happy to take any questions. Either one.
Can you kind of compare, maybe I missed it, but compare and contrast some of the study populations in which people have looked at neurogranin? I'm confused about what's control, what's prodromal, and what's normal aging, right? So you have a sample that is collected from Wisconsin that has some aging markers in it. So why is that not prodromal, or how is that different from other people's?
Yeah, it is confusing, and people use different terminology. So this group, so what I consider, I would consider this group an at-risk group for Alzheimer's disease because they have these elevated levels of family history of Alzheimer's and the ApoE4 gene, meaning they're at risk to develop it. If we look at their brains, they probably have a little bit more amyloid than the typical non-family history person. But clinically, as they present to us, they don't have any symptoms of Alzheimer's disease. So they,
It could be memory if you're...
Exactly. And so they could be in the prodrome. You could call them that, theoretically.
Can you call them prodrome at that point? Or
Some people would. I wouldn't.
like, there's other
Yeah.
studies that the controls, do they also have an increased neurogranin or no?
So,
Controls that don't have an ApoE4?
yeah, that's a good question. So I didn't parse it out by ApoE4 status. There was no association between ApoE4 and neurogranin. And the previous studies that have looked at neurogranin, they've just looked at controls, regardless of if they have a risk of Alzheimer's disease, compared to people who have mild cognitive impairment, which is really like the online prodrome, and then Alzheimer's disease. So they've shown differences among those. So we don't know if at-risk people necessarily have a higher levels of neurogranin than not at-risk people. I tried to parse that out in this group and didn't find anything, but the sample sizes were a little bit small. Yeah.
Could you say a little bit about assessing memory? And as a scientist, what you think about using scale scores? I'm assuming most of your data is cross-sectional, point in time, scale scores for memory. Is that right, first of all? And what I'm leading up to is the question of whether by, scale scores are adjusted for age and maybe education, trying to account for the variability you see in individuals throughout their life. And whether we might get much more subtle, detailed findings if we could use raw scores across time.
I completely agree. If you're looking at a longitudinal study of neuropsychological abilities, you should use raw scores. You should not correct for anything. And so we have larger cohort studies at the MAC through Joel's work. We actually don't see huge changes in memory in our older folks across time. It's more processing speed and executive functioning. But I completely agree, longitudinal work should use raw scores. If you're looking at cross-sectional, however, and you're trying to capture, what is different than expectations, I think using age-corrected or demographically corrected scores can really help capture that. Part of the reason why I used an age-corrected score, and it didn't matter if I used it both raw and age-corrected in this study, but it just allowed for fewer covariates that had to go into the model. So you're already adjusting for an important factor that I was going to adjust for anyway. But it's a great point, well taken. Yeah.
Thanks. Really interesting work and really exciting. So As a generic kind of thing, not knowing anything about your work, anytime I see a lecture on biomarkers, sort of a list of things sort of pop up, right? So, and particularly when you have surprising results, that my first inclination would not be to go to a complex biological explanation, would be just kind of run through the list of problematic aspects of doing biomarker work, so state versus trait, hidden compounds and variables, problems with the study design, those kinds of things. What is your assay like? Could this be technical error? So I'm wondering, you know this work better than anyone, so can you kind of give us a rundown? What are the challenges with measuring neurogranin and CSF? What are the potential compounds in the study that would be maybe an alternative explanation to sort of the complex biology, both with regard to that sort of increase at the end where you have these outliers, but also it strikes me that the finding with regard to ApoE is really surprising, that the entire construct is around sort of the relationship between neurogranin as a marker of kind of overall synaptic health. You've got a group that's massively increased in risk over population baseline. So is that a power issue? Is that, so maybe if you can just tell us, if you were writing the end of a paper that said the pitfalls to this study would be, before we moved on to claiming that this is a reliable biomarker, these are the ones and this is what we've ruled out as technical, blah, blah, blah.
Yeah, no, I think you raised a great point, and I definitely meant to mention, but I think that sort of unusual quadratic curve needs to be replicated. We're really not sure what's happening there. It could be completely spurious. Some of the factors that go into measuring these proteins are as nuanced as how long the CSF has thawed for, the room temperature, the time from the antibody insertion. I mean, there's so many very nuanced factors that go into what comes out at the end. And you raised a great question about state versus trait. I'm super interested in what that is. Are these markers of some sort of neural reserve? When we think about cognitive reserve, what is expectations at a cross-sectional level? What do we expect for just a normal what levels do we expect a normal person to have? I think measuring these markers across time will be incredibly important. Again, speaking to memory abilities too, you need to be able to see relative changes. That's something I hope to do.
So do you know about inter-individual variability and like diurnal variation or anything like that, or this is
no.
so new?
Yeah. I mean, short answer, no. I'm proposing a grant that I can hopefully study it longitudinally. But yeah, it just hasn't been looked at. So that's one of the challenges of writing a grant about something we don't know anything about. So, yeah. Any other questions? Great. Thank you.